Hi, hey, hello everyone. This is the this is the topic. This is a lecture. This is a video for the lecture topic number 6 about DNA. And in this topic, I have two major things. One is the DNA structure, and the second is how the DNA is replicated. DNA structure itself is very important because after the DNA structure has been discovered, is people become certain that DNA is a genetic material. And next, more efforts has been focused on DNA, how to base on DNA to develop new technology to have a better treatment of the diseases, have a better outcome, agriculture, etc., etc. So let's first focus on the DNA structure. A DNA structure has a DNA has the monomer of DNA is called a nucleotide. A nucleotide itself has uh, three components. The very center, this is a ribosome. It's the first, second, two, three, four, and then five carbon. And this, the fifth carbon is bind to a phosphate, and this the first carbon is bind to a base. I don't know how to draw a base. And then there are the DNA and the RNA is only different in the second carbon. In the case of RNA, the second carbon has an OH group or hydroxy group. In the case of DNA, it has only a hydrogen. And this is important for the difference of DNA and RNA because since its H is smaller, two DNA nucleotides, when they join together, they can be very tightly to bind to each other. And then the base, in both cases of DNA and RNA, have four different bases. In the case of DNA, you have A, T, C, and G. In the case of RNA, you have A, U, C, and G. The full name, adenine, thymine, cytokine, and guanine. But you know, actually know the full name. Okay, it is good enough. And then these two, those kind of nitrogenous bases, and each of those A and T both can form two hydrogen. So an A nucleotide can potentially form two hydrogen bond to bind to a T hydrogen, T nucleic and then C and G can force three hydrogen bonds to bind each other. That's the basic pairing rule of the DNA. And then two nucleotides can join together. How two nucleotides join together? It's really hard to join. This is 3 prime, this is 5 prime, 5 carbon, and have phosphate, and then 3 prime can form a covalent bind with the with the 5 prime carbon of the next nucleic acid. And then you can form this one. Keep going, keep going. You keep going by 3, keep going, and then now you have a strand. We call it DNA strand. And this DNA strand have a direction. This is called 5 prime and this is called 3 prime. And normally we say this is the head and this is the tail or end. When we read the DNA sequence, how is the DNA sequence? For example, if this one is A, this is a T, this is a C, and the sequence is A, T, C. So DNA sequence is basically the sequence of different bases reading from 5 prime end to the 3 prime end. And this we call a single strand DNA sequence or ssDNA. And ssDNA can potentially bind with another DNA, and when these two DNA bind to each other, the other band will be exactly match, or we call it complementary matching the previous band. This is called a double strand, dsDNA. And this dsDNA, as we see, this is like a letter, but in the real world, it's not a letter. It's more like double helix. Okay, that's better. Yeah. So this is called a double helix DNA. A double helix DNA is the 3D structure of how DNA exists in the cells. And so basically, a key thing about double helix is that there are two strands, and these two strands is has opposite direction, and it's called anti-parallel. Anti-parallel means first, the two DNA strand has an opposite direction, and also the distance between these two DNA strands are constantly the same along the whole DNA strand. Anti means opposite direction, parallel means the distance is the same. And then between the two strands, this DNA nucleotides always pair with A to T and C to G. And then in this and in the whole strand, these two strands are not evenly distributed. You can see the gap between the two strands. Here is a small gap, means minor groove, and this is a big gap, a major groove. And you can see there's a lot of the DNA double helix model outside, and how you can tell whether this organization or whoever draw this is professional. If they draw a minor, and a major, is professional. If all the gap are the same, yeah, amateur. So this is the structure of the DNA, and then why people realize DNA can be the genetic material after find out the DNA structure, because a DNA can be extremely long, very long DNA. Sorry. And then this extremely long DNA with a sequence of the ATC, G, whatever, which you can try. With this sequence in their DNA, the DNA sequence can provide the required information of the genetic material. What is required as a genetic material? There's two things. First, it need to be able to encode the genetic, the sophisticated information, encode information. It needs to have some form of the structure can encode information, and this information is in the DNA sequence. And then secondly, DNA genetic material stay relatively stable from generation to generation. For example, my parents, they have two eyes. Both have two arms and two legs, and I still have two eyes, two arms, two legs. The major features didn't change, don't change from generation to generation, which means genetic information stays stable from generation to generation. And then it means genetic material need to be replicated or duplicated precisely from generation to generation. And then how the DNA can be duplicated precisely, that's the second topic of today. Okay. Let's see how. So, how the DNA can be replicated. Now you have a very long DNA molecules in the cells. How you can replicate this DNA molecule in the cells. At the beginning, when DNA is a double helix, it's extremely stable. Nothing will change. No DNA mutation will happen when the DNA is stable, act as a double strand. DNA mutation only happens when DNA is being replicated. And first, how you replicate it? You have to open up. You have to open up the double strand. So this is called melting. Melt. Is that right? Melting. Something wrong. Never mind. Melting the DNA double strand. And here at the center, this is called a replication origin. And through the replication origin, the replication will go this way, to all the way down. And then this is the frontier of the DNA replication, called the replication fork. And this replication is done. And now it's opening. It's done by this called a polymerase, DNA polymerase 3. DNA polymerase 3 can make new DNA strand based on the sequence of the template. So this one is a template. The DNA polymerase will make a new DNA based on template. But however, if you only have one single strand of DNA template, the DNA polymerase can do nothing because it cannot bind to a single DNA strand. I didn't know where to start. To start the DNA replication, you have to have something already exist, and this we call it a primer. A primer and the strand and the template strand DNA form a partial double strand DNA, and then the DNA polymerase 3 will come to the partial strand DNA, and then it's always add a nucleotide to the 3' end to make it going forward. And in the cells, how to make it happen, this primer is added by something called a primase. Primase add a primer, and then the polymerase extend the primer going forward. And the primer, this in the cell is the RNA. Later, this RNA will be removed. How to remove it, we'll talk about later. And then, so look at this one. Here, in this case, this is a 5. In this is 5 prime. Let's see this one. All right, let me open a new one. Okay, and then there's another one. All right. This is origin. This is origin. So when you add a primer here, this is a 5 prime. This is 3 prime. You can extend the DNA to a 3 prime, but of this one, and this we call it a leading strand. A leading strand is that it's the direction of this strand exactly from 5 prime to 3 prime as the direction from the origin to the fork. However, how you can make this one, or down here, this is the 5 prime, 3 prime. This is also the leading strand here. And then how to make this one? How to make this one? You cannot extend any of the DNA strand that is to the 5 prime end. You can only add it to the 3 prime end. Now, how you can do this one? In this case, you have to open up the DNA long enough, and then you add a primer here. The primase another primer here, and then now in this case, this is 5 prime, the 3 prime. The DNA polymerase 3 will add here, down, and then back to the origin. And then when you open up furthermore, another primer is added here, and then going forward, DNA polymerase 3 add here, and then it reach here. Now it's still here. This is RNA primer. This is another RNA primer. In here, the DNA polymerase 3 will get off from the cells and then came to the polymerase 1. Polymerase 1's its function is to cutting off the RNA primer and replaced by DNA for the go. And then when it come here, now here is no longer RNA, it's a DNA fragment. This is another DNA fragment. RNA polymerase 1 can no longer work. Now you have the ligase. The ligase will not cut or make anything. It's only there's a one DNA strand, another DNA strand, only connect together. They call it ligase. And then this thing is synthesized, lagging behind the leading strand. It's called a lagging strand. All right. And then the lagging strand, you can see it need the DNA strand to be opened up. It need a special long single strand to make a lagging. And then how about in the end of the DNA molecule? In human DNA, it's a linear. At the end of here, so see, if this is the lagging strand. If you do not have enough space for the primase to come, then this is missing. In each round of DNA replication, the lagging strand is there's a little shorter of lagging strand for each run of a DNA replication. And then when the lagging strand is missing, this leading strand, since there's a single strand DNA, will be removed later. So which means after each run of DNA replication, the DNA molecule becomes a little shorter. And it sounds scary, right? Then in that case, after several rounds of DNA replication, the DNA may be shorter and shorter. And how the cells fix this one? And then at the end of this sequence, there's a special sequence called telomere. Telomere is the cap of the DNA molecule at the end of each DNA. And telomere is a repeated sequence. What do you mean by repeated sequence? For example, ATC, 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 ATC. So this is the elements of the repeated. And this ATC is repeated hundreds of thousands of times. I'm not saying that telomere has ATC. What the sequence of telomere you can search up. So in these cases, again, in most of the cells, telomere is being shorter and 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 shorter after each round of a DNA replication. At some point, the telomere is become so short. When a short telomere, a cell with short telomere, no more DNA replication and no more cell division. This is one of the ways to control that a cells cannot divide in unlimited times. However, in some cells, like stem cells, they do need to replicate many times. And how come the stem cell can replicate more times? There's another thing called telomerase. The telomerase is enzyme that can bind to the telomere to extend the telomere. And the telomerase have an RNA molecule inside that serve as a template. So we use a RNA template to extend the DNA molecule. And since the telomere is a repeated sequence, the telomere just add the repeated sequence again. You use the same template, same RNA template because it's all the same sequence. And what can I say? So in this case, in the stem cells, we have telomerase. Telomerase in the stem cells extend the telomere so stem cells can further divide with non-stop. This is a good thing. What's a bad thing? Cancer. In many cancer cells, more than 90% of cancer cells also have active telomerase, which make the cancer cell immortal. There's a bad thing about telomerase. So next, lastly, let's talk about what's about the DNA mutation. Let's talking about the cancer cells. What causes cancer? I think we mentioned before that cancer is mainly caused by DNA mutation. And what causes DNA mutation? Firstly, polymerase 3, DNA polymerase 3. It itself has a proofreading function, which means once it adds a nucleotide, it double check whether the nucleotide fit to the template. If it doesn't fit, it cut it off, make a new one. And by this proofreading function, the polymerase, but this proofreading function is not perfect. So there's about 1 out of 10 to the 8th order of time that the polymerase 3 can make a mistake. It sounds like a very low rate, but however, think about a human being. Each human cells have 3 billion nucleotides in one cell, and then if follow this rate, about 30 DNA replication error will happen. This number, if they accumulate it, it'd be a big issue. So how the cells fix it? In the cells, there is another system called a DNA repair system. Firstly, whenever there's no match, there's a bump here. This is a bump. So because these two nuclei doesn't match, and then some protein will recognize the bump, and then recruit other proteins. Other proteins come to cut it off, and then it leaves something like this. It's a gap. And then DNA polymerase 1 will come to fix the gap. And then the second fixation, normally there won't be no error anymore. And then one more thing, how did this enzyme know where to cut? That's because all the DNA has another thing called DNA methylation. All the DNA has a DNA methylation modification. And then at this point, when the DNA is only freshly synthesized, the new strand does not have any methylation modification yet. And that's why how the DNA repair system can know which one is the new strand and to cut a new strand only. But there's too many DNA error, or the cell's health condition is low, the DNA repair system is not in a good condition, some of those error may escape the fixing. And then they will even go to the next round of cell division. When they go to the next round of cell division, there'll be no more mismatch anymore. For example, here we have a mismatch here. It's a T, it's a G, okay? And then if you go to the next round of cell division, it's TA, and then it's C and G. Suppose the T is the original good sequence and G is the mutation, and then in the next round of cell division, the perfect match. There's no way any DNA repair system can find out this is a mismatch, this is wrong, and this will stay in the cell forever, and this become a mutation. Basically, a mutation is a DNA replication error that does not fix in time and stay permanently in the cells. This we call it a DNA mutation. And DNA mutation may be good or may be bad. But the bad thing, DNA mutation may cause many different diseases, including cancer, but not only cancer. For example, sickle cell anemia is caused by DNA mutation. But also, in the way of evolution, DNA mutation also provide the essential motivation for the evolution. Here in the section, I may make an example is that we humans and other apes can see 3 colors, but most other mammals only see 2 colors. And why we see 3 colors? That's one of the genes in our body got a mutation. So that is all for today for the topic of DNA. See you next time.